All right, well, thank you all for coming again to today's Grounds on the Go. This is again the seminar series that the Applied Research Institute and the UVA Research Park put on here in the park, probably twice a semester. And so this will mark the hiatus point for the summer, and then we will be back again in the fall with two new series seminars for the fall and then two in the spring. So keep an eye out for that, and if you haven't left your email out there, put your email on the sheet so Katie can keep you abreast of what's going to be coming in the fall. Okay, so today we're going to hear a super cool talk about how James Lander's lab has taken what is actually a really complicated forensic process for all of you CSI and NCIS viewers, and put it onto what is the same size as what we all, if we're old enough, remember a CD disc in the Sony Discman players that we used to walk around with. So this is really fascinating research, and so we're very pleased to have James here. I'm just going to take a few minutes to introduce him and then let him tell you about his great work. So James Landers received his Bachelor of Science degree in biochemistry with a minor in biomedicine at the University of Guelph, Ontario, Canada in 1984. And his PhD in biochemistry in 1988, after a postdoctoral fellowship at the Banting Institute at the University of Toronto School of Medicine, and as a Canadian Medical Research Council Fellow, he was trained in cancer biology and diagnostics under Dr. Thomas Spelsberg, an endocrinologist at the Mayo. He founded and directed the clinical capillary electrophoresis facility in lab medicine and pathology at the Mayo, and developed clinical assays using CE technology. While an assistant professor at Pittsburgh, he forayed into analytical microfluidic systems with the goal of developing the next generation of molecular diagnostics. His program moved to UVA, where they developed new methods for cost-effective fabrication for microfluidic chips for rapid prototyping of micro devices for chemical, biochemical, and genetic analysis. He has published more than 250 papers in peer-reviewed journals, 22 book chapters, and edited 3 editions of the CRC Press Handbook of Capillary Electrophoresis. He was the recipient of the 2008 Association for Lab Automation Innovative Technology of the Year. He was a speaker at the Nobel Symposium 162 on Microfluidics in Stockholm, and has been named to the Magnificent 10's Analytical Science Power List in 2017. So it's great to have you here, James, and thank you so much. And take it away.
Well, thank you, Mel. It's a pleasure to give you a little bit of information about the type of work that we've been doing. The title actually came from Mel, and I think it's an apt title because what I want to talk about is shrinking things down, trying to do what people have been trying to do for 35 years, and that is shrink the processes that occur on laboratory benches down into something that can potentially be handheld. And so with that, it's important to kind of draw on some of the messages that we've received from nature. And we'll talk about fundamentals of microfluidics, not to bore you to tears, but to touch on the things that are important and we can relate to. First thing is the driving force. And while this is not exactly where we're going, it's one of the things that has motivated my research program for a number of years. And that is, this came out of Peter Gascoigne at the MD Anderson Cancer Center at Baylor School of Medicine. And he put up these stats that essentially says 6 billion people in the world, we're going to need diagnostic screening, if there's residual disease, follow-on testing, pharmacogenomics, therapeutic drug monitoring. If you now tap into that number of pathogens that we know exist, number of species of fungi, all with the potential to become drug resistant, which now raises a completely new question about genetic testing, and then add to that testing of food, water, agricultural. He estimates this, that at some point in the very near future, and maybe even now, there's going to be a need to do 16 billion molecular diagnostic tests per year. And what he proposed when he presented this a decade ago is that the technology doesn't exist to do this. So how do you address that? And the answer is microfluidics. It's one of the answers. So what is microfluidics? There's a couple of different definitions. One is that it's dealing with very, very, very small volumes in very, very small spaces on devices that ultimately should be the size of a credit card. But it also is an interesting arena because it's at the cross-section of a number of fields: chemistry, biochemistry, clinical chemistry, surface chemistry, material science, engineering, all of with the focus of taking conventional methodologies that are now used in labs and trying to make them more efficient. So what's the message? Well, if you take a look at the evolution of microelectronics, the chips that we have in our phones and our computers are nothing more than three-dimensional metal superhighways for electrons. And you can conduct electrons with great control in terms of where they go and when they go through this very complicated architecture, and this is what the Pentium chip is. Now, it turns out that this is an interesting analogy primarily because it sets the stage for microfluidics, which most people don't really understand what it is. So this is nothing more than purified sand that essentially in crystalline form manages to give us now these silicon chips that do the processing with power that is 5 orders of magnitude more powerful than we've seen in the last 30 years. So let's just touch on that. So if you take a look at World War II and microelectronics, this is really the genesis of the need for better electronics. This idea of using vacuum tubes eventually became a limitation for the Air Force in terms of putting all the electronics that they needed into a cockpit. And so it was really back in the late 40s, early 50s that they started funding Shockley and a number of others at Stanford to start developing now microelectronics. Get rid of vacuum tubes, move to transistors, and now make simple calculations simple. And if you take a look at the system, my battery's dead. Do you have a laser pointer? So I can go without it. If you take a look at what was the state of the art in the 50s, it was an IBM calculator that literally took up an entire room. And if you take a look at what its capabilities were in terms of instructions per second, it was 50. Right? So in other words, this had less power than the very first calculators that came out from Texas Instruments. And if you want to now put that in perspective, compare the processing power with what is now ancient, an iPhone 6, and the comparison is 25,000. So that's the difference simply in terms of processing power. That doesn't take into account the differential in size. So if you take a look at that, here's the IBM 650 magnetic drum calculator back in the 50s. It was capable of doing 17,000 instructions per second. A lot of this was used initially for ballistics calculations. And if you now take a look at what is archaic technology now, which is the Pentium 4 dual core 2000, it's 1,000,000 times faster in terms of processing speed. It's 10 to the 5 times smaller. And so if you take a look at the evolution in terms of processing power relative to size, you've got 11 orders of magnitude in terms of improvement. And the reason I pose this is because the same question has been asked for microfluidics. If you now take large labs that are filled with instrumentation, is it possible to follow the same model as electronic microminiaturization and do that for microfluidics? Now, for those of you who know transistors, I pose a question. Anybody have any idea how many transistors are microminiaturized onto that Pentium chip? Anybody want to take a guess? Nobody? How many? 1,000? Deborah says 1,000. It's actually 42 million. Right? She said thousands of millions. Yes. So that should give you a sense for the ability to compress footprint without losing functionality. So what is microfluidics? Microfluidics now does the same type of thing. Instead of now having a 3D superhighway of metal for conducting electrons, you have a 3D highway of channels to move fluids. And if you can move fluids, you can do chemistry. You can meter, you can pipette, you can react, you can heat, you can do all of the things that we presumably can do on a bench. And if you take a look at this video, which came out of Steve Quake's lab at Stanford, Steve set this up as something that could now repeatedly meter very small volumes and move them into its separate location for some kind of analysis or measurement. And the interesting thing about it is that volume that is circled there essentially represents 100 picoliters. Okay? Not 100 microliters, not 100 nanoliters, but 100 picoliters. So it now gets you down into a regime that is way beyond micro. You're now into the nano and pico regime, which is exactly where you want to be if you want to have massive integration at the same time that you have massive footprint compression. So really the difference between microelectronics and microfluidics is nothing more than electronics versus fluids. And the microfluidic chips that we talk about are really nothing more than a chemistry version of the Pentium chip. Now, when we talk about going microscale, what does that mean? So there's a 6-inch ruler, and if we look at the metric scale, there's a millimeter. That's a millimeter. There's the 50 by 50 micron channel that we routinely put onto microfluidic chips to do separations. So that gives you a sense for the scale down. And just to give you a kind of a more representative, something you relate to, that diameter is roughly the diameter of a human hair. So you're talking about now taking fluidics that occurs in columns, and if you're a chemical engineer, in pipes, and now shrinking them down to this scale. So one of the things becomes critical as you shrink down are really two things. The first one is that as you shrink down the volume of a chamber, so if we were to shrink down this room to the size of a shoebox, nothing changes other than the number of molecules that are now in that box, because it's essentially a small part of what's in this room. But the thing that really matters is the amount of surface area in that shoebox relative to the volume is large. So as you shrink down, the surface area to volume ratio changes, and that's bad from a microfluidic point of view because things stick to walls. So one of the challenges you have is that. The other challenge you have is something called laminar flow, and it's both a blessing and a curse. So laminar flow is this trade-off between inertial forces and viscous forces, and essentially says that there's an equation, and that's the equation, that looks at the density of the fluid, the viscosity of the fluid, the size of the pipe, and the length of the pipe that's being used, and asks whether or not the flow of fluid through that pipe is actually turbulent. In other words, is it mixing? So that's the equation, and to take a look at this in a different way, there's the Reynolds number equation. So the Reynolds number essentially tells you with a single dimensionless number whether or not the flow in that feature is going to be turbulent or not. So let's take an example here. If we take a look at water, and in particular now here, look at the dynamic viscosity of water. Water's not very viscous. And you plug that into the equation, you essentially get a Reynolds number that's around 15,000. Now, according to the chart on the right, if the Reynolds number is less than 2100, you have laminar flow, in other words, non-turbulent flow. If the number is greater than 2100 up to about 4000, it's in this transition region, but eventually it transitions into a turbulent flow, where now as you flow through the channel or through the pipe, you now have a lot of mixing. Now let's compare this to corn syrup. So corn syrup now has a very, very, very different dynamic viscosity, not surprising. And if you now crunch through the numbers, the Reynolds number is actually 4.7. Substantially less than 2100. Therefore, it indicates that the Reynolds number is low. Therefore, the flow through that viscous medium is not going to have any turbulence. And there's an interesting video that actually demonstrates this. So this is corn syrup. And now this individual is going to take 3 different dyes and very carefully place them in that corn syrup. And inside that little cylinder of corn syrup, there's a paddle, and after they load the 3 dyes, you're going to see that they'll very slowly turn the paddle to essentially mix those 3 dyes. And so now he's going to rotate it 5 times. And as he rotates, you see that you start to see a smearing of those dyes. And the question becomes is if this is really laminar flow, if this is really non-turbulent flow, after he spins this 5 times and presumably have mixed the 3 dyes, if you now reverse that, you should actually get back to the same spot that you started. So you're going to see now that he's going to start reversing this. And now he'll reverse this with 5 turns of that. I actually tried to figure out how to show a video at 2x, and I showed just how useless I am with technology because I couldn't figure out how to do it. But after 5 turns, watch what he ends up with, because it really is a perfect example of low Reynolds number. So in fact, in that viscous medium, there isn't an opportunity, even with some mixing, to actually cause turbulent flow and mix those dyes. So it's a really interesting illustration. But we're not interested in working in corn syrup because there's not a lot of applications for that. But what we are interested in is shrinking down pipe sizes. So if you take a look now at the same equation, and this time use a pipe that has a radius of 1/10 of a meter, and run through the calculation, you get a number, a Reynolds number that is 928,000. Turbulent or non-turbulent? Tim? Turbulent. Now if you do the same calculation and now take the diameter of that pipe down to 100 microns, 1/10 of a millimeter, and do the same calculation, you now get 928. So it's in that transition that we start to see that, in fact, the same kind of fluid with the same viscosity in 2 pipes of a different size actually transition from turbulent to non-turbulent flow. And that's an important thing for microfluidics, because what it says is that we can now take 3 different solutions, have them come in from a trifurcated structure into a single channel, and they will remain essentially non-mixed in a laminar flow as it progresses down that channel. And if you were to watch this in real time and follow some particular volume as it progresses down the channel, you would see eventually at the interface you start to see a little mixing. But that mixing is only diffusion based. It has nothing to do with turbulence in the channel. In other words, microfluidics gives us the opportunity to now mix things and not have them mix. That can be a blessing, but what if you now have 2 reagents and you want to mix them? Then that's a curse. So, interesting phenomenon. It's a physical limitation that we have to deal with. Now, if the mixing at the interface between 2 different streams is driven by the diffusion constant, there's the equation for the diffusion constant. And let's take a look at 3 very different sized particles. We'll look at a potassium ion. Potassium ion has a size of about 0.3 nanometers, so very small. And if you take a look at how far it will travel just by diffusion, 1 micron, 100 microns, 10 millimeters, and 1 meter, it's a small molecule, its diffusion constant is large, and therefore it diffuses very fast. So in something that is a standard channel in a microfluidic system, we could now transition from one side of a channel to the other in about 2.5 or 0.25 milliseconds, or sorry, 2.5 seconds. Now if you take a look at a protein size of roughly 3 nanometers, so now orders of magnitude larger, and if you take a look at now in that same channel how quickly that protein diffuses, it's much slower because it's a bigger molecule. And then if you now were to go and break open a cell and take something like a mitochondrion, an organelle, and essentially take a look at that, the size of a mitochondrion from end to end is about 2 microns. And if you now take a look at how long it would take for that to diffuse from one side to the other, it's actually 3 hours. If you want to have that mitochondria diffuse from over the space of about a meter, it would take actually 30,000 years. So this idea of diffusion in laminar flow can actually be exploited. It means that things won't mix, but depending on the analyte, you actually have the ability to have something change from one side to the other. So here's an example, and these are 2-micron particles. This comes from Mike Ramsey at Oak Ridge National Labs. He's bringing flow in from the top, flow in from the bottom, and then flow in from the left. And you'll see that those particles that come in a diameter, the width of the channel, essentially focus down into a very, very tight stream. In fact, they're single particles. So here, by doing nothing more than exploiting laminar flow, you've actually done something that's called electrokinetic or hydrodynamic focusing. It's pretty cool. Now, the other thing you can do with it is now flow one fluid in from the top on the left, one fluid in from the bottom on the right. They remain laminar over the course of the channel in the middle, but if those particles now have the appropriate diffusion coefficient, they can actually change sides. In other words, you can use this as a microfluidic laminar flow filter without any filter. Simply the fact that the particles are diffusing fast enough to cross over into the other stream allows you to now isolate those things from a mixture. So again, an advantage of laminar flow in this inability to mix. Now what happens when you want to mix? Because if we're going to do chemistry by adding multiple reagents, you've got to mix things up. George Whitesides and a number of other people, Howard Stone at Harvard, have shown that you can now put structures into the channel that actually cause, in some physically predictable way, a mixing of the fluid. And so one of these things is to put in these chevrons, these herringbone structures. So if you're traveling down this channel, there would be a series of these barriers in here that you would have to go over. And because they're chevron shaped, they cause some very, very funky physics that allow for now mixing of those components. So despite the fact that the flow here is actually laminar, you can actually completely mix something from one end to the other in a very, very short distance. In fact, Carmen Reedy, who worked in my lab a decade ago, did some very interesting stuff with this, putting these types of structures into glass microchips. So what does that all mean? What's important when it comes to microfluidics? Well, all of these things. So is the surface water-loving or water-hating? Is the surface smooth or rough? Is it flat? Is it nanostructured? Inert, reactive? You don't want things to react with the surface because that's really bad in microfluidic systems. Is it transparent or opaque? Can you actually shine a laser through and detect on the other side? Is it conductive? Is it insulating? Biocompatible, denaturing? And if you're going to work with biofluids, this one turns out to be really important. You don't want to put a protein DNA sample into the microfluidic device and have it glom onto the walls and never ever come out. So biocompatibility is important. What's interesting are these, and there's some interesting demonstrations of this, and I'll show you a couple. So first of all, why is it that the water strider doesn't sink? Well, it's a surface tension issue. And they understand with great detail the structure of the leg, the angle of that leg, and how it now plays into the surface tension on the surface of water so that it essentially can stride on top of the water. On the bottom you see a nanoporous surface where they now have the ability to essentially electro-wet and bring up droplets and have them form on the surface in real time. And so now you're controlling nanofluid and even picoliter fluids and controlling where they are and what they do. Now what does it mean to be hydrophobic or super hydrophobic? This is a video that shows this. So this is out of a colleague's lab at Denmark Technical University, where they put onto this surface an incredibly, incredibly hydrophobic material. And you'll see that the water not only will not sit on the surface, the energy is such that it actually rejects the surface and essentially pops back up. So these are the type of tricks that you can play, and this is a valuable piece of information because how the fluids interact with the surface of a channel in a microfluidic device is important. So labonachip is not this. But 1988, Andreas Montz essentially suggested that if we take a glass microscope slide and etch a channel in it and cover it with another glass microscope slide, we can create a microfluidic channel that allows us to do chemistry. So this idea has been around for an awful long time. This is actually the 1990 paper. And what's important are the applications. And he had identified back then that this concept of micro-total analysis systems, in other words, not an analytical device that does one thing, but an analytical device that does all of the things you need to do in a lab to process a sample. And so this bridges analytical chemistry, diagnostics, food environment, all of those sub-disciplines where analytical or measurement science is important. So why go there? Well, there's a number of reasons. If you shrink down a bench onto something the size of a credit card, or as Dr. Henriksen said, a disk, you now have the ability to do the same chemistry with much, much, much less reagent. That's valuable. You have the ability to do it with much lower volumes of sample. Depending on what the application is, that may or may not be important, but here's one example. When I was at Mayo, we developed a test in a microregime type of analysis system for viral encephalitis. And the sample for that was essentially a spinal tap. So these patients, they think they've got viral encephalitis. They have a ticking clock to determine what the virus is so that they can hit it with the appropriate antiviral. So they do a spinal tap, and if they don't get a full mil of cerebral spinal fluid, they can't do the assay. And often you would talk to the docs and they would go, I can give you 100 microliters. And in a microfluidic system, that's more than adequate. So it's in those kinds of scenarios where the volume of sample is important. What else is important? It's got to be rapid. And as we'll talk about today, portability is important, and automation. And these are just a couple of examples that are now old that talk about the type of things that people started doing on either glass or silicon or polymeric-based devices. So, cool stuff. I actually gave Mel a different title for the talk, and she chose not to use it. It was cool microfluidic blank, blank, blank. And this is one of the examples of cool stuff. So this is recently out of the literature. These guys have used now microfluidics to essentially print skin grafts. So very much the way that you would use the old tape-based whiteout, where you'd slide the tape across the word, same type of thing, and you just pull a trigger and it essentially lays down a skin graft on an open wound. And this is going to revolutionize things certainly in the military, but any type of rapid response where you now to very, very quickly need to seal off something so that you don't have a chance of infection. So that's just absolutely mind-blowing. And at the core of it is microfluidics. This one where they're using droplet microfluidics and a silicone type micro device to essentially exploit how E. coli produce certain pharmaceuticals. And so now in one device, you essentially can figure out how to genetically engineer one strain of E. coli so that they produce in high efficiency the type of pharmaceutical that you're interested in. This is one now that dovetails into precision medicine, where now inside of a droplet, they'll put 100 or 1,000 cancer cells from the tumor of the very patient who provided that sample, and now do drug testing in the device to figure out which cocktail of chemotherapeutic agents actually work. I mean, that's the definition of precision medicine. Don't just do a carte blanche, we're going to hit you with this triad cocktail that works on adenocarcinoma 35% of the time. We're actually going to try a series of 20 drugs, and we're going to see which ones kill those tumor cells that came from you. And that's where it's going. They're already doing this at Johns Hopkins. So where does this all kind of funnel into something that's relevant to what we're interested in and also what Dr. Bienvenu and Dr. Root are very interested in, and that is, how do you exploit this to do things like Craig Venter did in terms of sequencing the genome? So Craig Venter sequenced the genome by using microscale technology. He actually used 300 of these and managed to sequence the genome in 3 years, despite the fact that Francis Collins had actually been at it with NIH sequencing centers for 7 years and still hadn't completed it yet. The new technology allowed them to expedite. Now, the not so good part of this is that Craig Venter, who was Caltech prof, very innovative, decided he was going to patent all of the sequences, so that anybody who developed an assay for a particular disease marker, they would have to license it. And then, of course, the government got involved and actually did the right thing and essentially prevented that. But the bottom line is it was microscale technology that allowed this to happen. So how does that fit into what we do? This is biometric analysis for human ID. There are a number of ways, and the current description is multimodal biometrics, where you use a number of these. But the question that's been asked more and more over the last decade is DNA really the ultimate biometric? And so I suggest, if you haven't seen this movie, that you watch this movie. So this is called Gattaca. I'm sure most of the women in the room will not argue against watching a movie that has Ethan Hawke in it, or Jude Law. And what in this movie they talk about, in this futuristic time, this ability to pick up a speck of dust and put it into this instrument and essentially sequence the genome, because dust is often just shed epithelial cells, and from that identify who was there. That's really not all that far-fetched. And so the question is it realistic to actually create the Gattaca box? And knowing for certain that it won't be that size, but can you create something that at least is benchtop, and then eventually something that's more portable? So let's figure out whether or not the microscale actually affects your ability to actually separate DNA. So this came out of Mike Ramsey's group, who's now at UNC. And Mike showed something very interesting here. He showed that if he input with an electric field a sample containing 2 dyes, and then electrophoresis those 2 dyes, as you would do in any kind of gel electrophoresis, what Mike was able to show was that he could separate those 2 dyes in a fraction of a second. So watch after the injection. How long does it take before you see 2 entities separated? So Deborah, I want you to count. Ready? Go. 1. So in a second, you've already resolved 2 chemicals that are distinct based on their charge. And Mike actually took it further. He actually created an incredibly small device where the dimension here is somewhere around 400 microns, so 4/10 of a millimeter. And what Mike showed is that he could actually separate those 2 things in less than a millisecond, not a second, but sub-millisecond separations, which says that despite the fact that you shrink things down, you still have the ability to obtain separation power, resolve 2 things that are different in structure. So how does that play out? Well, it plays out into microfluidic devices that now are trying to supplant what is done conventionally by gene sequencers. And one of the things that we did way back in the day, and you'll see that this involves the good Dr. Bienvenu here, who used this as a means to make an argument for actually getting out of her graduate program. And we showed that you could do this kind of integrated sample in, answer out analysis in less than a half hour. Now, if you talk to Joan, she'll tell you not nearly as pretty as it presents here. What's that? But the bottom line is it was one of the first examples to say that you could actually put a whole sample, and that sample was about 700 nanoliters of mouse blood, into the device and actually get a readout that said something was there. And so that was pretty powerful. Now, Fast forward, probably half a decade, and the good Dr. Root here, who was heading up a team at Microlab Diagnostics in Charlottesville, had shown that on this complicated but very elegant microfluidic device, you could do DNA separations that gave you the same kind of separation power that you could only get in a capillary that was 37 centimeters long. So what they had done that was so elegant was they had shrunk the length of that separation in order to get those DNA fragments separated from 37 to 7 without taking a hit in performance. This was an important development because it meant that there was something more important than simply the size of the channel and the length of the channel. So enter into the equation this concept of rapid DNA. 2008, the FBI launched this as the Andy program. They funded a couple of companies to build now integrated instruments that would do sample in, genetic profile out. In other words, they had the vision that this potentially could be used in booking stations. 2017, this was actually ushered in as an act by the guy from Utah. Thank you, Orrin Hatch. So it's now real. I mean, rapid DNA and potentially its use, very much like fingerprints are used, is real. So this was the scenario that would be involved with those kinds of analyses. Essentially take a cheek swab, run it on an integrated device, have it be automated, not require any skill, and then essentially get some kind of ID based on a database and have now a match-based decision. Now they set metrics for this, and the metrics were speed, cost per sample, and then this nebulous, it had to be affordable for a police station. Now, it was at that point when we were developing the technology at Microlab where Brian Root was the program manager, Joan was the program manager for Lockheed, and Lockheed had determined that the value or the cost to meet that metric would be the cost of a patrol car. So it had to be around $75,000. Because a police chief in a given year would make a decision, do I buy 4 patrol cars or do I buy 3 and a rapid system? So this is what eventually came out. These two instruments, one out of MIT, one out of Berkeley, we were out of UVA. Those two instruments came to commercialization, and they changed the footprint of DNA analysis for forensics. Now the DoD evaluated that technology, and I think the DoD had actually funded part of it, and they were interested in going from this to this. In other words, this idea of having a 2-man luggable instrument didn't meet their particular ConOps. So through the Applied Research Institute, we managed to get connected with the right people who brought in funding to have us now move from there to something that was backpack portable. So here's the evaluation they did as they surveyed what was available for that particular ConOp. There was a technology gap. They knew what they wanted, but the technology that was available was too costly. So these instruments were $250,000 and $275,000. The consumable was too costly because the cost per analysis was anywhere from $250 to $1,300. And so the technology solution that we pitched to them was get rid of that, put all of the chemistry onto this, and simplify the instrument. And so they bought into that, and we are now in the last 2 months of a 3-year project. So how did we take a new approach? We divided this into chipware, hardware, software, and chemware. And it's only these two that I think are worthy of talking about today. If you're going to change up the chip, you're going to have to make it inexpensive. So we actually fabricate these chips by using overhead transparencies because they are available or were available and inexpensive. We actually use relatively simplistic instrumentation to fabricate these. So we use a laser cutter, which is nothing more than a trophy etcher, and we use an office laminator. And if you now coat some of the sides of the microfluidic disc with toner, when you assemble that and put it through an office laminator, the toner melts and becomes a glue. So it allows you to do a relatively simple fabrication. So this is actually a 30-minute video that's sped up to about 90 seconds. And you can see that the first step is to essentially print the various layers. This is now the trophy etcher essentially cutting out the middle sections. And what you're going to see Yi Wen-du, who's making a 5-layer device. She's eventually going to take them, lay them out, orient them properly, stack them and align them, put them between a piece of paper, put it through a laminator, and lo and behold, what pops out is a microfluidic device. And the cost of that microfluidic device should be substantially less than $1. And the reason that is because graduate students don't cost a lot of money, and so they're cheap labor. But Dr. Root has actually done the evaluation in terms of what we think it will cost to have this mass produced. And we think it can be, certainly sub $10, maybe even sub $5. So I'm going to pass this around. This is one of the devices, and I've only brought one. But you can get a sense for what this is like. So why is it circular? Well, it's circular for a very good reason. We're going to use centrifugal force to essentially move fluids. In other words, what we want to be able to do is move fluids from the center to the periphery and control how fast they move, where they go, and do all of the things like pipetting and metering and mixing by doing nothing more than changing the spin speed. So why is that advantageous? Because it allows you to circumvent this. The vacuum pumps, the pressure pumps, the solenoids, the manifolds, all of the things that we built into the Microlab Lockheed instrument to essentially control moving very, very small volumes of fluid. And point in case here is that the Microlab instrument that we made was actually the lightest of the three. It was 110 pounds. But 30 pounds of that was essentially pumping hardware. So from the get-go, by going centrifugal, you essentially dump 1/3 of the total weight of the instrument. So that's moving in the right direction. Now, how do you control where things go? And we're not going to talk about this in detail, other than to say that if you create a stroboscopic video while the disc spins and monitor spin speed, you can see that you can control very accurately what flows where when based on spin speeds. So these are just two different ways to do that. And you'll see that as you increase the spin speed, eventually you break through some of those valves and you move fluids to where they need to go. And this is essentially the same volume of fluid being moved, but each one of these elliptical chambers represent 3, 2, 1 microliter, half a microliter, and then the one on the far left is 250 nanoliters. So not only are you controlling what goes where when, you're controlling how much. In other words, you're doing pipetting on a disc in a very, very simplistic way. Now it turns out that's not enough to do what we're trying to do on the DoD project, so we have to bring in now the use of physical valves. So these are things where we exploit the fact that toner on the chip is black, and if you hit it with a red or green laser, nothing more than a powerful laser pointer, you essentially will burn a hole, and you see it burning it there, and you will burn a hole in the surface, and you will connect essentially two channels that aren't connected. In other words, until you do that 1 or 2 second blast with that laser, you have not opened up the flow through that channel. So that allows you some fairly interesting capabilities. And it allows you now to start working on a much more complicated device. This one has 13 valves, where now you can control which of those valves get opened at which time to carry out these processes. And I don't want to belabor the details here, but it's a relatively complicated device. It's been designed primarily by Jingyi Li, who's part of the group, who's a great design guy. And it essentially cordons off now all of the things that you need to do on this device, everything from lysing the cells and getting the DNA out, to amplifying those DNA copies that tell you something about the individual, and then doing the electrophoresis. Here's a quick video that shows inserting the swab. So the person has already swabbed their cheek, and in a mere 10 seconds, you've now inserted that. So the whole simplicity that requires no skill is there right from the get-go in terms of the sample in part. In terms of doing PCR, this thermocycling mediated amplification of certain DNA sequences, this is a very, very old video, but it shows you, number one, the spinning, and the spinning moves the fluids to the right place. It'll also show you that in order to heat a part of that disc, you have to clamp down 2 heaters, and you're going to see now the clamping of a heater on top and a heater on bottom. And now those heaters will now cycle through different temperatures that allow you to amplify the DNA that give you the signature that you need. How am I doing on time, Mel? Better wrap up. So let me skip to what matters. So there's the conventional instrument on the top left that is used in forensic labs. Again, a 35 or 37 centimeter capillary. This is what, under the very careful tutelage of Dr. Root, who's a superb DNA separation scientist, that is the type of separation we can now achieve in 5 minutes. So again, not only shrinking the size, but shrinking the time. You can't have one without the other, otherwise you run into problems. And the trick happens to be this hydrophobically modified polymer that we add into that channel. Brian did not get out of his PhD program until he actually figured out how to synthesize that in Anneliese Barron's group at Northeastern. And it turns out to be probably, bar none, one of the best polymers out there, even relative to what's sold commercially by some of the big genetic analysis companies. So here's what this looks in just a quick video. You load the disc. Spinning now not only moves fluids, it actually helps us take care of bubbles, because getting bubbles into small chambers and small channels can be problematic, but if you spin hard enough, you remove those. There's laser alignment to now go and burn the valves, so you just saw one of the valves burn open. But the really interesting part is that because the disc is free moving, when it comes time to do the very last step, which is electrophoresis and DNA separation with fluorescence detection, the system has to find the channel. And this is something that Dan Nelson, who in our group has been really creative in terms of defining. We essentially have the optics that use that laser move, and you're going to see the laser comes on, and now the optics are going to scan back and forth and then change Z height until it finds dead center of the channel. And I think we can now do that in 90 seconds? Is that where he's at? About 90 seconds? So this is immensely important, because for those of you who know what a Sony Discman is, you'll know that if you've got a Sony Discman in the disc, or in the instrument, and you move it, the disc rotates freely. So unlike other microfluidic systems where the chip is hard mounted in one position, this was a critical development, and one of the things that the guys did that was incredibly impressive. So here's the instrument as it currently stands. It's nothing but a funky, complicated, slightly more expensive DVD player. You put a swab in, you insert the disc, you press go, and this is the type of profile that you would get. This is actually old data. This is going back to phase 2. But we're now currently getting that kind of data out in about 37 minutes. Now here's the important thing. That's the size of the instrument. Because one of the metrics that the DoD had laid down for us was this thing had to be a certain volume, fit in a backpack, but it had to be 10 pounds. And just to baseline for you how that ranks, remember that the 2 instruments that are out there commercially right now weigh 60 pounds for one of them, and the other one is 178 pounds. So this is an order of magnitude reduction in weight. You can run different people, and again, this is old data, but we can see the differences so that we can do human ID. We can still see that we've got a little bit of work to do with PCR, but it's the fluidics that we're now focusing on in terms of trimming that down. And one of the questions we've obtained from many people is, well, you can only do 1 sample per disc. So I went to Jingyi Li and I said, compress the architecture so that we actually have 2 swab chambers and we can do 2 per disc. And sure enough, he did. So we think there's a number of ways. One of the things that we're getting feedback on is, why does it have to be the size of a CD? Well, it has to be the size of a CD because you want it to be small and compact. The response is we don't care if it's small and compact because we're just going to sit it on a lab bench. Then you get back to the old 33 1/3 RPM records. Who in here knows what those are? Yeah, right? And they've asked the question, if you can put 8 on there, why not have a disc that's that big? And it's a reasonable question. So what do we know? We've developed a simple method for building these devices out of cheap materials. We can print valves. We can open those valves with a $9 laser. We can have multiple levels. One of the students has built an 11-layer, 9-fluidic level. So this is like a skyscraper where you can control chemistries in different levels. Cost possibly sub $5, but this is to be determined by Dr. Root. But this is the important one. It's 10 pounds and it's roughly the size of 2 reams of paper. And just to baseline that, this instrument, which is over at Microlab still, 110 pounds, 18 pounds of that 110 was cable. So we've now built an instrument that does the entire thing and weighs half as much as the cabling that is in the fully integrated system. So with that, these are the people who have been absolutely critical. We've got engineers, scientists, graduate students, postdocs, research scientists, even some undergrad computer science students, and they're the ones that do it. And you know, they're young, they're not jaded, they're energetic, they're not burnt out like I am. And if you just don't box them in, you'd be amazed at what they can do. So with that, I'm happy to take any questions.
I got one. So I probably mispronounced the company.
Theranos, yeah.
Was that based on Microfluidics? I realize it didn't work, but is that what they were going for?
Yes.
I've got plenty of comments on that, none of which I'm prepared to make on camera. But yeah, I mean, it was proposed they had the ultimate microfluidic solution, and I think what was eventually borne out was they didn't.
Right.
So, yeah, it baffled all of us. For those of us who've worked in this arena,
Right.
to have a chip that did 15 or 20 different tests off of a drop of blood, it didn't make physical or microfluidic sense. And I guess it doesn't.
Yeah, right.
The outcome.
Dr. Langers, can you
Yeah.
tell us a little bit about the STR chemistry that you guys are running on this instrument?
Yeah, so we've worked with kromega on, and it's just a 10-plex. We ran through the trade-off with having a very high plex, 18 or 24 loci, relative to the amount of time it would take to amplify. And our theory at that point was the PCR amplification was the biggest time sink. So if you trim back the loci, you should be able to do faster PCR. And they're currently doing 13-minute PCR, so you really can do that. But we also worked with them to make sure that the loci were less than 300 bases. So we didn't have to separate out to 600 bases. Again, so that the separation can be 5 minutes, not 15. So it's the standard loci, and I can share those with you. And they were chosen based on the random match probabilities, which one was going to give us the best discrimination. But we made the original mistake of doing the calculations based on Kromega's database on the American population, which was not the right calculation to do. So the random match probability, I think, 1 in 55 billion, something like that, which for their con ops is apparently adequate. And so that's what the customer needs, that's what you build.
So I'm curious, this is today, you talked about how long it took to get to today where you were. So 10 years forward, what's the future of?
That's a great question. So let's take it out of the military world for a second, right? Because if you think about, where did GPS come from? Well, GPS came from the military. They needed accuracy in ballistics. Where did microelectronics come? They needed to have smaller electronics to go into the cockpit of a plane. I mean, those are civilian, we've reaped benefits as civilians from military investment. And I think this is maybe a much smaller, but a similar scenario. So we're working with the Multi-Organ Transplant Center at the University of Toronto. And it's the largest multi-organ transplant center in North America. And what we think we can do is change the chemistry out so that series of peaks that look at 10 different parts of the genome, look at 10 different genes that they've identified that if they actually monitor those genes being expressed up and down, they can define very surgically when to pull a patient off of immunosuppression. Now, something I didn't realize, most people die post-transplant not from rejecting the organ, but because the immunosuppression is so toxic, it kills other organs. So the transplant docs walk this very, very fine line of, do I pull them off too soon, or do I leave them on because I pull them off soon because I don't want to, I think they're not going to reject the organ, or do I leave them longer because they're going to reject the organ, but I'm killing the liver now? And they walk that fine line constantly. So monitoring that panel of 10 genes, they've actually, the study that they're currently doing is 32 out of 33 have had successful allograft acceptance. I mean, that's mind-blowing. That's a game changer for transplants. So what they see potentially happening is you give the patient, when they go home, an instrument and a box of discs, and on a daily basis they do a needle stick, a drop of blood, run it, internet it back to the doc, and the doc monitors in real time how they're progressing. So if we accomplish nothing else other than to put that into this world, then I can die a happy man. So, and I think that's just one of a handful of offshoot applications. I mean, we haven't even scratched the surface. No other questions?
So what's the military using it for? Similar to police force to identify people?
They're using it like fingerprints.
Fingerprints. And is there an ocular version of doing it as well?
Yes, I'm being told yes by those who know. But that would require that they actually have an ocular database, right? So that.
Oh, yeah.
Right.
So do they have a DNA database of bad guys?
So I'm told.
Okay.
Well,
So is it going to cost what a cruiser costs, or is it going to cost what a police officer?
Dr. Root, what's it going to cost?
I think the company that licenses it gets to decide that, but they can probably spend more.
Yeah. I think it'd be 75K or less. And you know, if you go with the diagnostic model, right? All of the companies that make diagnostic tests, they actually give the instrument and they make their money. It's a razor and razor blades, and they make their money on the consumable. And you know, what these guys have managed to design might actually be cheap enough that you could actually consider that as a model, which is groundbreaking. Yeah.
What do you think are some of the issues related to this technology as it currently stands now? Like some of the lingering issues?
Well, trying to get it to work 100% time is an issue. There's a number of issues. What goes on that disc from the moment you put the swab in till the time that you get that genetic profile, 35 minutes later, is a 29-step process. So 29 steps that involve either moving fluid, mixing it with another fluid, heating it, cooling it, moving it into a different location, mixing it with some other things. So 29 steps. So first comment would be, there's plenty of places for things to go wrong. So, and I think what we've done, I guess, with Brian Root as the program manager is we've started to hone in on the technical part of it is pretty robust. You still have issues having to do biochemistry on a device. And think about that. If this is a chamber where you do PCR amplification, what you're looking at the wall is essentially a series like a multi-layer cake with toner in between or adhesive in between. And the questions that we've had to address over the years is, are those adhesives toxic to the chemistry that we're trying to do? Is there something that's happening in the very first step that is bleeding its way to the very last step and messing up the last step. So there's a lot of challenges in there. And so I would say at this point, they're material and chemistry related. The fluidics and the technical aspects and performance of the instruments actually looking pretty positive.
Do you have any idea on the shelf life? What kind of storage condition?
So the reagents that are going to be on there are polymer for the separation. That's Dr. Brute's polymer. We know in blisters that's pretty stable, right? Although we haven't done long-term storage on it. The PCR reagents are in a little candy floss ball, and the reagents that do the DNA extraction part is a liquid that is stable at room temperature for 2 years. So we've addressed all those things, but can I tell you that it can handle 0 to 40 C for 6 months? Not yet. I mean, we're going to have to do those studies. But those metrics have been presented to us. We know what we've got to hit.
Next phase, phase 4.
What's that? Phase 4. Yeah.
Organization stability.
Anybody else? That's pretty cool, I think.
You're welcome. Thank you.